Hello, everyone. Good evening to all. Namaste, and a very warm welcome to all our participants who have joined us live. Thank you for taking out your time from your busy schedule of either college or office, job, or anything. And I can assure you that you're investing your time in for a good cause. Okay. So I am Urmimala, your host for today's exciting session, and I can already feel the energy in the air. So yes, can I have with a show of hands here who all are excited to join this session and learn with our eminent speakers today? Yeah, can I have some comments from you all from the audience? A hi or anything like that? Show your presence so that we can begin with our session today. I hope I'm audible to everyone, right? Okay. Assuming that I am audible and visible to you all, I will be awaiting your response. So, our topic today is something, yes, very good evening, Sadaf. Yes, RVM Edit, Surya Kala, Banwari. Okay, I think that was slight delay in your comments. I'm receiving all your comments now. It's really nice to see all of you joining. Yes, Sai Charan, Sabiha, all of you, again, welcome to everyone. So, as I was saying, our topic today is one that will define the future of life science research. Yes, hello, Dr. Tahir Khan. Hello, Nikita. Good evening to all of you. So, the topic is NGS and Multiomics deep dive, advanced analytics for next gen research. Right now, let's pause for a moment and think. Imagine a world where just one drop of blood could reveal not just your current health, but also predict disease years before they happen. So it might sound futuristic to you now, but well, the future is already here, and it's powered by next gen sequencing and multi-omics integration. So it's extremely important to learn. Yes, hello, Jayant Kumar, Banwari Lal, Satyam. Thank you so much for all your responses. And yes, so from decoding entire genomes overnight to combining data from DNA, RNA, proteins, metabolites, modern research is now generating insights at a speed and scale which you have never seen before. But with this incredible power comes one very big question. How do we make analysis? How do we make sense out of this entire massive data? So that's where advanced analytics, AI, and computational tools are stepping in, and it is turning the raw sequencing data into discoveries that can transform many aspects: healthcare, agriculture, biotechnology, etc. So today we are going to explore exactly that, how NGS and multiomics are shaping the way research is done, and what skills you as students or emerging scientists, researchers, PhD scholars, you need to master to stay ahead. So before we begin, here's a very quick look at our session structure. We have three distinguished speakers with us today here. Each of them will be bringing a unique perspective. So first, we will explore NGS and advanced research, the technologies, workflows, and real world applications. Next, we will look into molecular diagnostics and clinical genomics, where sequencing meets precision medicine. And finally, we will dive into big data and computational omics, understanding how analytics and AI are unlocking hidden biological insights. So each session will be around 15-20 minutes and followed by a Q&A session. So make sure to note down your questions because we will not be able to take up any questions during the session. We would be doing that at the end of the webinar. Now is the time where we meet our brilliant speakers for today. So let me take the privilege of introducing them to you. Our first speaker is Dr. Bhupender Singh. So Dr. Bhupender Singh is
Hello,
a
everyone.
scientist. Hello, sir. Thank you for joining. Dr. Bhupender is a scientist and trainer at Biotechnica Infolabs currently with a PhD in bioinformatics from LPU, Lovely Professional University. He has worked at CSIR IMTech as project scientist and senior project associate, contributing to breakthroughs in machine learning, cheminformatics, and in silico drug discovery. With over 700 citations and collaborations across national and international institutes, he has an impressive track record in applying AI and computational tools to biological problems. So he will begin today's session by taking us through next-gen sequencing in advanced research. Our second speaker for the day is Mr. Suhas N. Ghorpade. Sir, you're on mute.
Hello, everyone. Good evening. Sorry for that.
Yes, thank you so much for
Yeah.
joining us here, sir. We're privileged to have Mr. Suhas with us here today, who is an accomplished scientist with 7 years of experience in molecular and genomic diagnostics, currently working as scientist at Trista Sciences Healthcare Global Enterprises in Bangalore. He is an expert in oncology-focused sequencing, cell-free DNA extraction, and HRD testing, with hands-on mastery in operating Illumina, NextSec 2000, and other cutting-edge platforms. Mr. Suhas has played a vital role in high-throughput NGS diagnostics and workflow innovation in cancer genomics. Today, sir will be sharing his experience with you, and we are looking forward to your session, sir.
Thank you
And
so
I'm
much.
sure, yes, and I'm sure you will take us deeper into the practical side of NGS and multiomics diagnostics, from the where it will transition from the lab to clinical applications.
Yes.
And finally, our third speaker for the day is our own very own Dr. Nilofer ma'am. I'm sure we have many fans of
Yes.
Dr. Nilofer ma'am here. Yes. So Dr. Nilofer is an who already knows her from our biotechnical classes. She's an accomplished bioinformatics researcher with a PhD specializing in big data analytics for canceromics from MIT Edith University. Her expertise spans genomics, proteomics, metabolomics, pharmacogenomics, and precision medicine with advanced skills in Python and R programming. So she has published multiple papers in international journals and holds an Indian patent for her innovative bioinformatics research. Ma'am will conclude the talks by guiding us through computational approaches in canceromics, biomarker discovery, and AI-driven analytics. So that's enough from my side. Without much further ado, let's begin our journey into the world of next-gen data and discovery. Can I see the Josh in our audience? Yes. Bring it up, bring it on. BIO. Can I see from the audience? Yes, then only our speakers will get the excitement to transfer the knowledge, transfer their experience, right? All right, so now I invite our first speaker, Dr. Bhupender Singh, to start with his talk on next gen sequencing in advanced research. Over to you, sir.
Uh, Thank you, Urmimala ma'am, for that kind introduction. And I welcome you all to this very exciting webinar on NGS and Multiomics deep dive. So I hope I'm audible and my presentation is visible to you guys. So without any
Yes,
further
sir.
delays,
It's audible
okay,
and it's visible also.
thank you for confirming. So without any further delay, let's start with the today's topic. So I'll be just guiding you through about my presentation in a sense of what all things I'll be covering up. So first of all, I'll be talking about evolution of NGS technologies. So how basically sequencing came into the picture or came into our knowledge, and then how we move towards its greater evolution, say first generation, second generation, and then third generation sequencing. And then afterwards, I'm going to talk about high throughput sequencing for multi-omics data sets. And then definitely as these sequencing based approaches are data intensive and these are data hungry approaches. So I'll be talking about some of the challenges which are associated with once you have received your data and how you can handle that data carefully. And finally, I'll be talking about a case study wherein NGS-based technology have been applied to a particular disease research, and I'll be discussing about how crucial that NGS-based technology was to actually progress or identify, say, clinical biomarkers for a particular disease. So this is my content for the today's presentation. So I hope you all guys will be follow along with me. So without any further delay, let's move on to evolution of NGS technologies. So the just historical background that Robert Hawley was the first, say, scientist who actually did a first nucleic acid sequencing. That was your alanine tRNA molecule, and it was done in 1964. All right, in from Saccharomyces cerevisiae, and then of course we have Frederick Sanger, who actually came up with the real breakthrough of sequencing by introducing chain termination based sequencing method, right? Then after that, later on, the first, what you say, first sequencer, automated sequencer machine, which was your applied biosystems, came into the picture, and it was launched in the United States. So if you are able to see this particular picture here, I have just tried to introduce or the basics of basic methodology behind their sequencing processes for all the 3 generation sequencing methods. So I'm just going to talk about a brief overview of how these technologies work. So starting off with the first generation sequencing, so that is referred to as your Sanger sequencing. So in this sequencing, we have, I mean, we isolate our genomic DNA of interest, and then afterwards, we basically fragment DNA into very smaller parts, and then, of course, cloning and amplification parts take place, and then we go for the sequencing. So now how sequencing happens in this? So in this particular part, sequencing basically happens based on your chain termination method, which is mentioned here. So what is chain termination? So this chain termination is basically based on the specialized type of what you say nucleotides called as DDNTPs. So are you guys aware of this term? I hope you guys are aware of this term called as DDNTP. So these are dideoxynucleotides, right? So they are responsible for actually stopping a reaction cycle. So so the main coming on to main agenda or like main process by which the sequencing completes. So they basically different types of this DDNTPs are fluorescently labeled, and they actually produce different chromatograms. So chromatogram means related to your color. So different color peaks are representing, as you can see here in this particular chromatogram. And basically this chromatogram is generated with the help of capillary electrophoresis. And then based on this characteristic chromatogram, you get your, distinction or finding what particular bases are present in our query DNA sequencing. So now moving ahead to the second generation scheme. So the process for second generation is quite similar till the what you say fragmentation part. But then after fragmentation, like you might be aware that second generation sequencing, you have the opportunity to go for what you say parallel sequencing or do massively you can do simultaneously for say thousands to millions of reads. And then in this we basically have what you call small molecules called as adapters, small DNA sequencing. Those adapters gets attached to both the ends of your fragmented DNA, and then once these are attached, they are actually passed on through your what you say flow cells or solid gel solid tube, in which you have a complementary adapters to the ones which are already attached, so which will actually helps in binding. So once the binding takes place, your PCR cycle basically starts. And then if I talk about briefly, the bridge amplification process starts, and then basically your single strand of DNA gets converted into double strand of DNA, and likewise you get multiple cycles and basically a cluster of all the a particular type of fragmenting DNA we are get. But how detection is done? So detection is basically done in different cycles. So cycle wise is, I mean, we basically go through cycle wise. So through each cycle, a different type of base, as you can see in this particular image, different type of bases are passed through, and based on their fluorescence, you will come to know that which particular bases are present at what location. Now, in contrast, when I moved on to third generation sequencing, as you can see at the very last end of this image, so in this is quite distinguished and I mean quite distinct from all other technologies because it does not require any PCR. So what we have in this we have a nanopore. So this nanopore, through this nanopore, we are actually continuously passing electrical signal. All right. So what happens when your DNA molecule, without any fragmentation, passes through this what you say nanopore? There is hindrance in electrical signal, and this hindrance, you can see a squiggling lines, and these squiggling lines are actually characteristic to a particular type of basis. So that's how, based on the presence of these squiggling lines, you get what you say characteristic lines, and these characteristic lines are basically representative of your any of the bases which are present in the DNA sequences. So this is a brief overview of how these technologies work. So now, likewise, I have told you that second generation sequencing, first generation sequencing was introduced by Sanger. It is particularly attributed to Sanger. And then in second generation, we have actually this ability to do what you say, sequence thousands to millions of DNA fragments. And then, of course, we have Illumina sequencing, which came into this particular sequencing methods, which can through which we can do whole genome sequencing and all other sorts of analysis. Then I discussed about the basics of third generation sequencing. So it was basically has the capability to do sequence much larger DNA fragments. And then examples, of course, we have PacBio sequencing and Oxford Nanopore sequencing. So these sequencers have the capability to do the sequencing of tens of kilobases in length. So just a quick comparison, not comparison or keywords, I can say, how these technologies differ from each other. So based on read type, you can clearly see that Illumina is short read sequencing, and whereas PacBio and Nanopore is our long read sequencing. And the sequencing technology, what is the basis behind them? So Illumina and PacBio generally uses sequencing by synthesis, whereas your Nanopore sequencing goes through electrical impedance. Now, talking about the most crucial part, which is your read length. I mean, for a single run, what is the length of a DNA fragment which gets sequenced? So you can clearly see that nanopore DNA sequencing has the highest number of what you say bases or a single strand which can be sequenced at a particular point of time. So now moving ahead, so we have high throughput sequencing. So if I want to introduce you to high throughput sequencing, so it was basically what you say a paradigm shift from what you say you have one tradition that one size fits all. So maybe if I talk about this about in the terms of drug discovery, so we have a single type of medicine, say for a particular disease, for all the patients who are suffering from that disease. So this high throughput sequencing actually provides a paradigm shift from this to say a personalized medicine or precision medicine. So in other words, what I was going to talk about, that this high throughput sequencing is basically providing us the capability to multiomics approaches or to multiomics analysis. So I'll be discussing about all these technologies in brief in the coming slides. So, but first and foremost, what are the benefits of this high throughput sequencing? So first and foremost, we have parallel sequencing. So we can analyze hundreds to millions of DNA or RNA molecules. And then, of course, they are quite fast in comparison to the older generation or previous generation methods. And then finally, efficiency, they are also efficacious enough to actually process larger data sets, and the user can do multiple comprehensive analysis through this, what you say, platform. Now, this was the part which I was talking about, that what all versions which we have, which we can opt for based on our need or based on the problem which we have in our hand. So likewise, the very first, which is very common and known one, is our whole genome sequencing, in which we directly go for our sequencing the entire genome. So wherein it can help us. So obviously it helps us in, if you are interested in some drug discovery projects, or we want to identify predisposition, or we want to introduce personalized treatments. So now, what are the advantages, say, for industries? So yes, we can get a comprehensive genomic view, and also we can go for, there is a possibility, not a possibility, directly we can identify the resistance mechanisms associated with a particular, say, antibiotic. So the next type of flavor which we have in high throughput sequencing is RNA sequencing. So this is the most popular one, as per my, what you say, knowledge or experience. So this, in RNA-seq, you basically look for the expression of particular gene, right? You go for gene expression analysis. Then the main idea behind this RNA-seq is we actually want to identify or figure out what all sorts of genes, or what all sorts of genes which are actually regulating in their expression, say whether those genes are overexpressing or underexpressing in a particular condition, say any diseased condition, in comparison to the healthier cohorts, right? So this basically helps us to identify, say, drug targets, or again drug resistance mechanisms. So this sequencing also a very quite popular one, and this has actually shifted the dynamics of, say, drug discovery. Now, at last, we have this AT-AC type of sequencing, so which is basically a step back to start with your, what you say, central dogma. So prior to gene, what you say, transcription, then we have gene regulation mechanisms, right? So through this type of sequencing, we can actually go for epigenome profiling. So in what we do in this, so in this epigenome profiling, we actually know that what regulatory mechanisms are associated with a particular condition, say, like I have already mentioned, say for healthier ones or diseased ones. So through which we can identify that what all signatures are distinguished in, say, any diseased condition, and through these analysis we can actually help us in finding out any biomarkers for a particular disease. So this is a basic overview for all the high throughput sequencing approaches which are available for multi-omics data analysis. Now, moving ahead with our NGS data challenges. So, at this stage will come like when you have collected your sample, you have done your sequencing, and now you have the actual sequence data in your hand, right? So this is where the bioinformatics begin. So the very first challenge which will be having is your volume and scale, right? So in which you definitely might be having a very high amount of data, say in GBs of data. Maybe you're working with n number of samples, thousands or hundreds of samples, and there can be issues for computational resource, how you will handle that data, and there can be other IO bottlenecks. All these things actually are quite difficult, and if they are not addressed properly, they can actually cost you too much, and there can be a significant data loss, and then of course you can have long run times. But obviously we have some approaches, like we can go for what you say high performance computing or computing over the cloud, or we have various compressed data. We can go for data compression, like we have BAM format. So this is a binary alignment and map formats, in which your alignment from your reads to the reference is basically converted into a binary format, which is less taking less storage, and it is more easily readable by the system. So then again, how you can mitigate this? More options are, like you can also go for development of your personalized workflow, or you can go for pipeline optimization based on your needs. So now moving on to next challenge, which is basically your data quality. So this is a very crucial challenge. I mean, we might be having base calling errors. Base calling error in the sense maybe due to some contamination or some errors in your sequencing procedures. You might be getting, say, detected T instead of A, right? Or any other sort of contaminations like adapter contamination. So this can actually lead to false positive or negatives, right? While you will be calling for variants, say maybe identifying variants for disease X, for cancer, what all variants are there. So if you have these sorts of what you say impurities or contamination in your what you say sample, that may lead to biased results, which can actually lead to what you say improper results, or say false positive or negative results. But how we can mitigate those? So definitely we have some standardized tools, like you might be aware about. We have fast QC, then we have trimming the adapters, and maybe removing or filtering the reads by their quality, like, and removing duplicates. And then finally, last but not the least, we have data pre-processing challenges. Maybe we have to decide on various cutoffs, or there, I mean, as per my personal experience, so there are, say, tens or hundreds of tools which are available there to do a single analysis. Say you want to do mappa, you want to what you say, you want to align your reads to your reference. So maybe you have different sorts of molecules and these different sorts of tools, and these tools works on entirely different procedures, and they have very different criteria to choose that how they will assign a particular read as a hit to your reference. And based on that, their analysis or their inferences will change. So this can actually lead to if you I mean, this is my personal experience. We have, I mean, I where I've worked previously, we have used different pipelines, say for bacterial genome annotations, and different pipelines were giving us different genomic reads, and these different genomic reads were actually getting annotated to different gene names, or say protein names, or any gene ontology. So the I mean, as per my experience, one way I can suggest is to use standard tools, or maybe you can actually search in literature and look for the tools which are actually what you say peer reviewed, and those are published in a reputed journal. So this is the one way that how you can mitigate or minimize those challenges. So now coming to my last part, so which is a case study. Like I've told you, I'll be discussing about one case study in which basically NGS has changed the face of your what you say for disease research. So this paper is actually published in Nature in 2020. So this is basically from UK Biobank's data. So what they have done in they have recruited hundred thousands of what you say participants were recruited, and their consent was taken, and like their blood sample was taken, and then WGS was performed with 30x what you say coverage. So this study was basically to identify what you say variants or variations or single nucleotide polymorphisms in your for rare diseases. All right. So once that sample was taken, whole genome sequencing was performed, and then all the bioinformatic work started, including calling the variants and prioritizing those variants. And then there were two steps. It was bifurcating from there that part, and then first was that they have found diagnostic grade gene. So diagnostic grade gene means if that they have got enough evidence on their hand based on this variant call, that gene is actually corresponding to a clinical report or a clinical diagnostic, right? So this MDT is basically your multidisciplinary team. So this multidisciplinary team was actually verifying that whether that particular variant can be established as an clinical indicator of or clinical variant of a particular disease. So if they have identified enough evidences, this was actually reported to the clinician, and then that clinician uses this information to improve the health of that patient. But secondly, if they don't found what you say enough evidence, then they went for association analysis, in which they identified that maybe in those hundred of thousands of sequencing, what all variants were actually associated with particular rare diseases. And those studies were, if they identified particular signatures, then those signatures were tested in lab conditions. And likewise here you can see that if for a particular gene, you can see that the expression maybe in healthier ones is lower, but maybe in diseased person its expression is higher. So that's how they came to analysis, or they came to an inference that this disease or this particular marker might be contributing to a particular clinical case. So likewise, say they have done some analysis, like they have identified what all clinical phenotypes can be attributed to various type of disease conditions. And these maps were actually representing that what is the frequency of those phenotypes in particular disease conditions. And most of these diseases are actually including your hereditary diseases and all those things. And next was to find out the importance, this plot was actually telling you what all clinical features are found in maybe say different types of diseases. I mean, how many types of diseases a particular phenotype is contributing to. So likewise, this study was done, and then they have identified, likewise here you can say that for this particular instance, if I can talk about this IRD. So this IRD basically report to your inherited retinal disease. So you can see that in 53% of the cases, this IRD was found to be associated with all these all these type of ailments or rare disorders. And then they also plotted this one plot. This was actually telling the gene rank significance in the sense that what all genes, maybe if what is the possibility of a single gene which is present in multiple reports or multiple type of these phenotypes or say disorders, right? So that's how this analysis was done. But the whole idea here was, you can see here, you can appreciate this, that through whole genome sequencing, a single sequencing, they were able to identify, they are able to develop a clinical markers, and they also identified new markers which were characteristic of rare disease. So through this, I hope you can actually go through this paper through this PubMed website. And this paper will actually help you appreciate that what all wonders this NGS can do, and how NGS actually is shaping the healthcare future. So with that note, I would like to sign off. That's all from my end, and I wish you all the best for your endeavors. Thank you very much.
Wow. Thank you so much, Dr. Bhupender, for that incredible and insightful session. The way you explained the evolution of NGS technologies from first gen to next gen sequencing highlighted all the benefits and challenges involved in data interpretation, and tied it to the real world disease research, the case study especially. It was truly fascinating, and I'm sure our audience gained a much clearer understanding of how NGS is driving modern discoveries in both academia and industry. This is the future, guys. And if you have any questions for Dr. Bhupender, please post it in the comment section, and we will be taking it up during the Q&A session, right? So thank you so much, Dr. Bhupender. We'll see you again during the Q&A. Yes. So with that, we are moving on to our second session now. We are quite excited for this. I welcome on stage Mr. Suhas, sir, for your second session.
Thank you, Urmimala. So I'll
Yes,
just share my presentation. Just a minute. Good to go.
Sir, can you please make it
Yeah.
full screen?
Yeah, just 1 minute.
Sure, sir.
Okay.
Yes, sir. Thank you.
Thank you. So can I start now, Urmimala?
Yes, sir. Please, sir.
Yeah, thank you. So good evening, everyone. I hope the day was good for my fellow researchers, workers, and aspiring students, and anyone who just stumbled onto this. I hope you get to learn to something. So the topic I'll be covering today will be multiomics integration and system biologies. So let's get into it. So this will start with what Dr. Bhupender and Urmimala also pointed out. This is the era of multiomics approach. There are always been eras of different things. Like if you're a sports fan, there was a Ronaldinho Ronaldo era, there was a Messi Ronaldo era, there was a Tendulkar era, now a Virat Kohli era. If you're a movies fan, then maybe there was a Ashiki era, and for the Gen Z people out there, now a Sayyara era. But what I'm getting at to it is every field has an era, and in the field of science and especially NGS, this is now the era of multiomics. So starting, like Dr. Bhupender also pointed out, we the basis of this comes from the central dogma. So there is the DNA to RNA to protein to metabolite flow, which is unidirectional. But in this, we have the different kind of omics. So omics essentially comes from the word meaning whole or group, and that is the origin of system biology also, which is just a holistic approach, taking the individual aspects and putting it all together. So let's say when you talk about genomic, it's from the DNA. What are the SNVs, copy number variations that we find in the DNA? When it comes to epigenomics, it might be methylation, how the DNA is getting methylated at certain positions. For transcription or transcriptomics, you might look into the coding, non-coding, or RNA fusions. The proteomics, as the name says, the proteins and metabolites is finally the phenotype. So essentially, the multiomics is when you combine all of this together. So that forms the basis of getting a holistic picture, looking at it from a wider lens instead of looking at it. And there's a benefit for both. Sometimes you have to look very specifically, sometimes you have to look from a wide angle, and multiomics allows you to do that. Once you get a particular data from the special scope, you can broaden out and use it as multiomics. So it's a very, very layered technology. There are three different approaches you can take for understanding the multiomics. So you can go the genome first, where you're looking in the genomics, like the previous slide, genomics, epigenomics, transcriptomics. You can go the phenotype first, where you're coming from the phenotype, like in some diseases. When you go to a doctor, they see the phenotype first, and then they get into the genomics. So that will be your starting point, and it could be in from the environment factor, even though it's not explaining here, and everything is interlinked, as we know from the epigenome. If you see the red dot, that might be one particular instance where this particular gene is flowing into the transcriptome, it's getting into an RNA, and then it's getting into the proteome, it's connected to two different proteins, and that's how it works. It is very, very interconnected, and you can imagine on this on a very, very large scale. So the approach can be different, the starting point can be different, but ultimately you will end up in a position where you might need all the data to get a clearer picture. So when you talk about getting so much data, there's also, like Dr. Bhupender said, there's also the part of integrating the data. So when you come to this, this is the key four pillars that I've understood that helps in the multiomics data integration. It is the conceptual integration, the model-based integration, statistical integration, networks, and pathway data integration. Actually, I do want to say that most of this will be pictures. I'm a strong believer in a picture is worth 1,000 words. So most of these are going to be photos, and I have linked the papers. These are very nice papers you can read in anyone who's interested, as references below. So coming back to the overview, so these are the four pillars that essentially the integration part of the multiomics work on. So we'll go one by one. Even you can read it on the screen, but I'll just try to put my spin and explanation on it. So if you look at conceptual integration, it's about using the existing knowledge and databases, like the Human Genome Project, which was started in the 90s, got finished in the 2003, and different databases that is from there which has come up based on cancer, based on neurological diseases, Alzheimer's, oncology. So you have, in conceptual integration, you take the existing data and look at it, where does your data which is flowing from the new experiment fitting into. Then you can go into the statistical integration, where you can take statistical techniques like regressive place, where you are predicting, like in drug discovery, you predict how the drug will react in a particular organ. Or it can be a comparative analysis, where you're comparing the relationship between two different proteins maybe. So there's a pattern or a systematic way in which you go for statistical integration. Then you come for the model based integration. There you put your mathematical and computational skills to use, and you simulate the biological system. It is useful in, again, in drug discovery or in protein protein interaction pathways, if you want to understand how the system as a whole works and not the DNA or the RNA. It's like the entire system. It's a holistic way to look at it. And then the final way is to networks and pathways. So this, you look into, the various interaction between the molecules inside the biological system. So there's the protein protein interaction that I was talking about, and you'll have the, if in terms of drug, you have the pharmacodynamics and the biochemical pathways, end stop gap codons, which come into play, and you understand that. So you basically understand the network, the biochemical pathways, and the structure with the biological system as a whole. So these are the four paths that you generally have when you are looking into analyzing the multiomic data. So these are certain examples of available software. Some are commonly available as free, but some might be paid versions. But all of them actually fall under the umbrellas of the four integrations. Like when you look at similarity and correlation, those softwares are working on the statistical integration way. The network and Bayesian will come under your network and pathways integration. Your fusion will be your model based, and your multivariate will be the final one, which will be your statistical no sorry yeah, sorry, even I got confused because it's kind of out of my domain, but I'm getting there. So the multivariate is your model based approach. So basically these four pathways, and I like the names that bioinformaticians comes for these, like the NEMO is one. So that is the neighborhood based multiomics clustering. And you have paradigm, which is pathway recognition algorithm using data integration on genomic models. So all of this helps you combine the eons of data that you have into a systematic way and looking at it. And these are the tools which will be commonly available. Some might be paid, some you might have access, but these are the four pillars which will help you understand how the data flowing from different ways can be integrated, and you need to get. And if you look at the picture, there's a color coding. So the disease, this is in reference to oncology, since that's my field. So there is the disease subtyping. So the blues are the softwares which are helping in your disease subtyping. The oranges are the ones which are helping you into disease insights. Like if you take look into paradigm, it does a bit of both. It helps in your subtyping and your insights. And finally, the brown is the one which will help you in your biomarker prediction. So these are the final tools which will help you integrate all the data that you're generating from the multiomics. Now coming to the real world applications of the multiomics approach. So this is my rosy roti, or rather bread and butter, precision medicine. So I've been working in this field for almost 7 years now. I started in the molecular field. Now I've come to the genomics, the next generation sequencing. And it is very, very essential when it comes to something. I'm speaking from personal experience here. When it's oncology, because cancer is ever evolving. So it's sometimes very, very, very important to have personalized medicine, because like a example is in lung cancers, a known target for drugs is called EGFR gene. So if you give EGFR gene throughout, it might not help all the people with lung cancers. You need to undergo testing to see if there is a mutation in the EGFR. There are certain mutations in the EGFR which help the patient absorb the antibody or the drug better. And there are certain mutations which are resistive in nature. So even if you provide the drug, it will not help the patient. And this is where, and when it comes to cancer or anything, it is especially cancers, the drug, the cost of it is very high. So it's essential that you look into it in a personalized manner, because ultimately there's a cost involved, and also the efficacy of the drug will go down if that particular treatable gene is not available. So this slide essentially is telling you what till now has been doing, like the multiomics is coming into the data integration, the statistical analysis, the visualization, and biological insights. What are the challenges? So handling data heterogeneity. Sometimes you don't know if the data coming from the transcriptomics and genomics might clash with other, or might not make sense. So, but it is up to your knowledge and how you know and have a deep understanding or look into it to understand the data which is coming might not be significant right now, but might work as a subset or a cohort for the future. So like in diagnostics, when we do the testing, we have a tier system of giving the genes. It's called tier 1, tier 2, tier 3. So the tier 1s are the genes which have a specific target, a drug known. So if that is present, the clinician will look into it and they will use that particular drug. Tier 2 will be something, the genes which are not commonly seen in some cancers, but it's an uncommon gene in another cancer. So they can maybe try, like it's like a EGFR maybe coming if in a colon cancer. Then you can try the EGFR gene in place of a colon cancer, but it is meant for the lung cancer. And then you have something called tier 3 or VUS, where you don't know the significance of the gene, but it is not known right now. It is called a variance of unidentified significance right now, but it can become a significant variant in the future. That's why it's important to keep testing and keep looking at it from different angles, because with the knowledge today can become an important tool tomorrow. So again, a real world application from precision medicine is plants. Plants, again, what we can do is we can look into the opportunities when we have, because agriculturally there's a lot of, there's a demand for the food is growing so high with our increasing population. It is critical that we get the genomics and the multiomics in it to meet that. When it comes to food, let's say cotton, BT cotton was a great example of it, where science met the agriculture uh, department, and they created something which helped everyone. So this is again a slide which looks into the different time points in where you can look into enhancing the plant physiology, or maybe yield the improvement or the nutritional causes, or some might just look into having a taller plant, it might help them. So there is a thing. So it multiomics has application in different fields, from precision medicines to agriculture, and it's important knowing where you can use it and how you can use it. So it is key that we keep on working through it, like the human, and everything takes time and effort, and it increases, like it the time shortens with the new technologies. Like if you take the human genome project for example, it started in the 90s and it took them 14 years to get to the first human genome project to be published. From there, 2004, the reference genome that was published, the next one, which is commonly used in NGS for data analysis and everything, is called HG90. That was published in 2009, just 5 years. Then in the 4 years later, they published a newer version, which was the HG38. So that the time always keeps reducing based on technology, and it is worth our time and effort to invest into multiomics, because it will clear so many pathways. Like personalized medicine, again it's more towards what I know. So personalized medicine will help everyone in general. It will be like you will be given the drugs which will help you. It will not be, you will be not treated as a whole, but as an individual. So with this, I come to the end of my talk. Hopefully everyone liked it, and we'll get to the end of it when we have questions. Thank you so much.
Thanks a lot, Mr.
Yeah,
Suhas, sir, for giving us
thank
such
you.
a valuable real world perspective. And that is straight from your Rosy Roti Diagnostics
Yeah,
Labs. And your
absolutely.
insights on, yes, and your insights on NGS workflows and the advanced sequencing platform, you truly connected the theory to application. And with the images, with the pictures, with the visual representation, I'm sure our audience were able to relate better and easy to grasp. So, especially how the clinical genomics is transforming cancer diagnostics, and your emphasis on precision, quality control, and all that, it definitely gave our audience a good glimpse of what cutting edge sequencing actually looks like in practice. So that was an eye opener for anyone who is aspiring to work in this particular field. So thank you once again for your enlightening session,
Thank
and
you.
we will see you. Yes,
Yeah.
sir. And we will see you in the Q&A session then. With that, we will now move on to our third speaker and our last speaker for the day. I invite Dr. Nilofer ma'am to kindly take over the controls for the third session. And a gentle reminder to everyone: if you have any questions, kindly post it in the Q&A in the comment section so that we can take it up in our Q&A session. Yes, ma'am. Thank you, and over to you.
Sure, ma'am. Thank you. I hope my audio is clear to all of you. And
Yes, ma'am.
yes,
Audio
ma'am.
is clear.
Great.
Audio is clear, ma'am.
So, okay, ma'am. So I will be sharing the screen. Share screen. Yeah, the slides are moving.
No, ma'am. I think you
No,
need to share the full screen
entire screen. Okay,
presentation mode. Presentation mode, ma'am.
sure.
PowerPoint presentation mode. Yeah. So, how has been the session so far for our audiences? Exciting things you're getting to learn today, right? All of you?
Yes, ma'am. The
Yes.
slides are moving.
Yes, sir. Yes, ma'am. Good to go.
Okay. So I hope that whatever the couple of sessions is covered so far, you guys get a very good deep overview regarding multiomics and its application for the same. Whereas today I'm going to like carry forward those information with the integration of artificial intelligence. Okay, because we know that as we are continuously trying to come up with the new technology, the new era, right? And if we are thinking in that particular perspective, okay, and we are looking for the biological data of our interest to perform certain kind of analysis and even the different predictions, right? Because if we were use the word prediction, so we will be thinking about what exactly the exciting things we can do with the integration of artificial intelligence, machine learning, and definitely followed with the deep learning context. So that is what exactly today we are going to walk through with the presentation. So all of you can see that on the screen, it's mentioned about the title, that is Advanced Analytics and AI for NGS, that is Next Generation Sequencing plus Multiomics data. So it is ultimately uncovering or else it is unlocking the biological insights with machine learning. So as you all know, guys, like we are continuously moving with the era for the big data comprehensive analysis, right? And if we just talk about the NGS data itself, right? So it's covering about like a GBs of data, terabytes of data. So it's really need to take care and understand that how we can overcome this challenge and we can proceed ahead with the analysis and interpretations of those particular data sets, because there are major applications of those particular topics such as NGS multi-omics approach, where we are able to identify certain biomarkers, we are able to identify the drug targets, we are able to uncover the biological mechanisms, its drug resistance overview, and many more things, right? So if that is the major applications while understanding the NGS or the multi-omics data, then definitely as a researcher, we really need to think about how we can integrate artificial intelligence, right? But before that, you guys really need to take care that you should know the basic pipeline or the basic overview, or as the concept at least, that what is NGS. Okay, because lots of researchers and the papers are even getting published based on the NGS pipeline multi-omics analysis, right? You will guys find a huge number of papers, research papers are get published with a very international journals accordingly, right? So if we are thinking in that particular context, okay, and we are looking to add our skills over there, or else we are trying to upgrade our skills over there, so we really need to clear with our concept, basic concepts about the pipeline or the workflow, right? Then only you guys will understand how to integrate the artificial intelligence technique. All of you getting my point, guys? Okay, so just think about that. Whenever you're trying to use AI, so remember that artificial intelligence is advanced field, right? So to come up with the integration, first we really need to know what is the basic pipeline we are looking for, so that AI can use to overcome the challenge, or else it can be used for the more predictive analysis. All of you agree with me? Yes, guys, can I get some responses? Yes, all of you agree? I hope so. Thank you. So you can see that basically this makes the overview about like artificial intelligence and multi-omics as a technology, which is considered as one of the revolutionizing field. And we are trying to understand this complex biological system so that we can target the disease mechanism or as the specific overall approach what we are looking for. With that context, as you all can see that if we are integrating the machine learning AI in omics data interpretation, so ultimately we, the researcher or the students who are there right now, okay, what are trying to understand this overall concept, so ultimately we are trying to learn the landscape. Okay, so what is now landscape? Landscape is nothing but where we are trying to break the omics data, that is multi-omics data. So within that, we have like a genomics data sets, right? We have the transcriptomics data, we have proteomics data, and many more omics approaches are there. And we are trying to integrate in a single overview and understand the overall approach. So ultimately, that concept is called multiomics, right? So as you all can see that genomic integrations is about all about like foundation of layer, where we are combining the different information about the genomics, trying to identify the structure variants, SNP kind of mutations, copy number of variations with respect to its genome sequencing. Okay, all of you, right? It's clear. Fine. So ultimately in genomics, we are trying to use the genome data. We are trying to come up with the more prescribed finding so that we can align with the genome reference, and then we can come up with a very prescribed finding. So if why to deal with like lots of multi-omics approaches, if that is your question, guys, so ultimately this kind of multi-omics approaches will help us to go inside deeper to understand the concept, or else to uncover the significant findings within the biological world. All of you are excited, right? So this is exactly what we can come up with the finding. And that's why rather than just stopping with only genomics overview, you guys can even move one step ahead to understand the transcriptomics data. Okay, so transcriptomics data is all deal with what is a gene and their expressions, how the differential expression genes are getting formed. Okay, and can we take those DEG or the differential expression genes to say that it's a significant genes or as a biomarker findings? So that is exactly what we can do with the transcriptomics data. And you guys can definitely see that right now we have number of pipelines available to process this transcriptomics data, such as the RNA sequence pipeline, bulk RNA sequencing, single RNA sequencing pipelines, right? And to help this analysis to uncover, we are also taking help of like coding languages such as Python and R for the same. Got it. So ultimately, we are trying to understand the gene expressions profiling. We are trying to identify the relevant pathways and the mechanisms which are responsible. Okay. So that is how we can use the transcriptomics and the genomics data to uncover this kind of findings. All of you are getting my point, right? So this is exactly what we need in today's era, where the one of the challenges about big data, raw data, how to do the curation, how to come up with the interpretations, right? How to come up with the findings. If that is your main questions, guys, so ultimately this multiomics pipeline will help you to answer that stuff. Getting my point? Okay, so then we have the proteomics data. So ultimately why we need to go deeper insights with the proteomics data, because proteomics is called as one of the quantitative and the qualitative informations is get shared in that. Okay, so ultimately we are trying to understand the biological informations based on its quality and the quantitative way. And if we are going deeper insights to come up with the findings, so ultimately we can work with the profiling. Okay, we can understand the molecular profiling, we can integrate the omics data with respect to its phenotypic condition, and then we can screen that kind of data sets with the personalized medicine concept. Exciting, guys. Okay, this is exactly what we can do with respect to the multi-omics landscape kind of analysis. Okay, I hope you all of you get my point. Whereas moving one step ahead with the next slide is that you all of you can see that the world of multi-omics. Okay, so basically this is exactly what is happening today in the biological world, right? So we are trying to use the multi-omics data, and it is called as the world of omics data, right? So why? Because the integration of genomics, transcriptomics, epigenomics, proteomics, metabolomics, right? Interactomics, transcriptomics, right? Whatever you guys can consider whenever you are trying to work on that concept. Okay, so ultimately it's providing us a tool, a new tools which are enables the scientist to acquire and analyze the data. Okay, and remember guys that whenever you guys are using multi-omics data, you can even pull out more than one disease type of your interest. Okay, and that is how it is called as the big data comprehensive analysis, right? And that will help us to understand how powerfully we can get a better insights to understand the mechanism and come up with the biological process understanding. So this makes a multi-layer data integration, okay, which reveals the complex disease mechanism that a single omics approach cannot capture. All of you getting my point, right? So whenever in future, if you are really looking ahead and you're planning, like you want to work on any project, or else you are really excited to go ahead and come up with any publication quality work, so just remember that if you go deeper insights with the multi-omics integration, so there will be high chances to uncover more significant finding with integrated approach. All of you agree? Yeah. So this is exactly, guys, what you can do, and you can really capture that if you stick with this kind of findings. Okay, so in that way we are trying to understand the mechanism, we are looking for the number of diseases to target. Okay, and that makes a research comprehensively. So the data exploration challenges are really there. Okay, I think in the previous session sir has mentioned about the challenges. So definitely the challenges are there, but we can definitely overcome the challenge because right now we have correct technology in our hand. Okay, so ultimately it is called as a right time with the right technology. And if that is the era where the right time and the right technology is dealing with that, so why not we will just deep dive with that, guys? All of you agree with me, right? So why can't we also in that race? Okay, we have kick up like we have started our career, so we'll stick with that, right? So that we can also understand how to capture this kind of findings. So definitely challenges are there, but the challenges can really overcome if we understand the basic pipeline and get to know how to overcome those kind of findings, right? So that is why you can see that the challenge with the data, with the big data, is nothing but scale of complexity. Okay, because the high throughput sequencing data such as the next generation Illumina technique, okay, 10x sequencing, nanopore sequencing, Oxford nanopore sequencing, okay, PacBio sequencing, so you guys can check it out. So you will find a lots of different experimental techniques are getting evolved. Okay, it's coming with a more upgraded kind of what you can say as a predictions, okay, with experimental stuff. And if we are able to get that right, so ultimately we are trying to work on very high throughput sequencing data which generates around 5 billion variants per long reads. Are you getting, guys? Okay, and that is against like whole genome sequencing of your interest. So the volume of data is really huge. And if we are trying to understand this multi-omics data with a high dimensionality, again, the one of the challenges heterogeneous data, okay, and the noisy data. So it really need to understand how sophisticatedly this can be get analyzed using the correct technology or the platform. And if I just give you the answer, guys, how you guys can overcome this challenge, so ultimately bioinformatics pipeline or the approach will help you to do that stuff. Okay, because bioinformatics is one of the field which integrate all this stuff for you. Okay, and virtually, guys, remember. Okay, so virtually you can sit, you can explore your data, come up with the analysis, do the findings, and then showcase your results accordingly. And that's why you can see that the analytic limitations are even there. Analytics means what is nothing but the experimental techniques, guys. Okay, so where the huge number of data is getting enumerated. Got it? Enumerations are nothing but the data is get captured and we are able to get the data maybe in the form of raw files, okay, like the NGS data is providing you the FASTA-Q files, right? Okay, BAM files are there, copy number of variations, variant files are there. So how to capture this data, how to analyze this data, that is also one of the challenge. But as I've mentioned that I have also given the answer for all of you, that we have the solution, we have the bioinformatics pipeline which help you to process your data in more smooth and significant way. Excited, guys? Okay, this is exactly what we can do. So that's why you have to even look about what are the challenges and the limitations, so that if you're aware about the challenges and the limitation, then only you're able to come up with like findings or solutions for that. All of you agree, right? If you know what is the challenge and the specific limitation, then only you will look or hunt for how we can look for the solutions. And that is how the traditional statistical methods are one of the challenge, okay, like multi-scale data integration, looking for the integrative approaches like the discovery context, right? How to come up with actionable insights, and the clinical interpretations is really important, guys, okay, where we are trying to integrate the genotypic data with the phenotypic information. And why to do that? Okay, because our ultimate aim is to identify and look for the precision medicine concept. Because as you all know that the today's era is depend about like big data and AI. So remember, guys, the future AI is depend on precision medicine. Okay, so all of we will talk about personalized medicine, precision medicine. So if you want to come up with that findings, okay, if you want to prepare yourself for that future research things, then this is a starting point, guys, what you can come up with, understanding the multiomics, AI, and all that kind of stuff. I hope that you all of you get the point, right? So the next slide is dealing with respect to the data exploration challenges, like multi-scale integration is there, as I mentioned, that combining the data with the level of view to understand. The actionable insights are there where the clinical interpretations can get added so that you can come up with a significant finding, right? You can look for the casual discoveries, nothing but identify the root cause of effect with the data. It's nothing but understand the mechanism, look for the specific resistance analysis, okay, identify the biomarkers, look for the fusion genes, okay, variants, SNP mutations, right, genetic mutations, okay, you can come up with those kind of findings accordingly. So in that context, multiomics integrations creates a comprehensive, guys, integrative pipeline, remember, and it is trying to uncover or reveals the interconnected biological networks. All of you are getting my point? Okay, so why we are trying to understand this multiomics, so that we can understand the interconnected biological networks or the mechanism, and the aim is to identify the therapeutic responses. It's nothing but biomarker discovery, drug discovery, right, identification of the specific mechanism, pathway, and so on. So in that context, if you just see, guys, multiomics integrations deals with genomics data, transcriptomics data, proteomics, metabolomics data, and if we are integrating this overall landscape, so ultimately we are trying to work with a network-based approach, means comprehensively we can work with the findings, and that makes the proper components and the techniques of multiomics to integrate in a such a way that we can come up with the potential findings. All of you agree with me? Yeah, this is exactly what you guys can do. So in that way, we are trying to understand how the omics approaches makes a proper comparison or as integration, so that we are able to understand many biological or as the multiomics techniques. Because if you just see, guys, like the genomics data itself deal with next generations, okay, whereas we are trying to come up with the proteomics, that is mass spectrometry data, right? Transcriptomics data dealing with the microarray data sets, okay, epigenomics dealing with bisulfite sequencing, cheap sequencing, right? And then the multiomics data dealing with LCMS, GCMS data. So every technique will have their specific approaches, or as the experimental analytical techniques are available. So every techniques will used for the specific processing, and the type of data which get available, like enumerated that data, will be get varies based on the specific data sets of your interest, okay. And that's why, guys, it's really challenging to process this data with the multiomics concept. All of you are getting the point, right? How why it is challenging? Because the genomics data will enumerate the specific data format. proteomics data will have the different file formats. So basically it's called as a multivariant file formats, right? So multivariant, why? Because every data will have that specific file format to process and analyze ahead. Because according to the experimental techniques, the data has enumerated. And we really need to take care of the data. Maybe it's a raw or else it's curated. We really need to process that data using the standard pipelines or the tools which are available in the bioinformatics platform. All of you are clear? Getting the point, right? So you are able to uncover those things, right? What I'm trying to explain. I hope that you all of you are getting some insights regarding the same, right? So next thing is that AI and machine learning. That is really excited, right? So AI and machine learning is ultimately where we are looking to look for like analytical game changer. Okay. So basically, why game changer? Because using AI and machine learning or deep learning, we are able to deep dive with the multiple data sets of our interest. Okay, the different features are there. Okay, the different factors are responsible, how to correlate the multiple factors accordingly, and then look for the proper decision making findings is nothing but data driven decision. All of you getting the point, right? So AI is directly taking your data and it is trying to give you a decision, and that is called as a predictive decision, prognostic analysis, predictive analysis, and so on. So ultimately, it's AI is uncovering the patterns within the quality data sets according to your interest, guys. And this makes nowadays, guys, remember that definitely we are continuously discussing about artificial intelligence, machine learning, and deep learning. But if you just observe, guys, that AI has moved one step ahead again. So we are using the automation processes, generative AI. Okay, so why these concepts are coming? Because AI is now enough. Okay, it has done very smart analysis. Now we are looking for how we can make automation processes, a generative AI process. Okay, where the multiple models is get stored and we will process our data according to those models, so that we will get one step ahead, a more predictive accuracy. Okay, this is exactly a real world, guys, where you will definitely work on the data which give you a driven decisions. Excited, guys? Okay, this is exactly what we can do. And that's why AI has make a separate power network analysis where the biomarker discovery can be get achieved. The one of the thing is the regulatory networks where AI will help us to uncover the complex interaction which are linking with the genes, RNA, proteins. Why? Because we are talking about the multiomics data. It is also helping with respect to identification of disease subtyping. For example, if you're trying to work on the breast cancer disease, right? So it contains the different types and subtypes. And every subtype is responsible for the specific resistance mechanism. That is what we are able to capture so far based on the research, right? Triple negative, HAI2, right? So it is very specifically showing the resistance mechanism irrespective towards its cohort population. And if that is our main challenge, guys, remember, then how this AI and ML can help us with that findings is nothing but it will help you to understand the subtype identification. What is the correlation? Okay, how we can come up with the alteration analysis where the molecular signature or multiomic signatures can be get captured, and we can work with respect to the very prescribed findings for our research discovery. And that's why we can even work with respect to the multiple variant at a time. Remember, guys, right? So for example, if you guys are trying to explore the gene and their expressions, right? So you will find that the thousands or lakhs of genes are getting expressed. So how you will try to correlate the expressions pattern, how you will get to know that which genes are highly expressed, which genes are low expressed, right? And if the genes are high expressed, again it's coming in the thousands, like 40,000 genes are get expressed, 50,000 genes are get expressed, right? Based on its expressions value. So how you guys think about you can come up with the biomarker discovery where you are trying to only showcase like 5 or 10 as a potential biomarker. Getting all of you? So coming from a big data where the lakhs of genes are getting expressed, then you are getting know that these are the top thousands of genes is getting expressed, and finally concluding the findings that it's a potential biomarker, maybe 2, 3, 4 accordingly. So that is exactly what the integration of AI and multiomics landscape will help you to capture this finding. Yes, guys? Are you really feeling excited? This is exactly what you can do. And you will definitely find lots of research papers are get published, right, with that context. So you will start correlating. So ultimately the AI has also have a specific role with AI predictive analysis in healthcare, like early decision. Okay, so why we are doing this stuff, guys? Okay, if that is your question, right? So ultimately we are trying to make this finding where the AI can use for the early detection. Okay, patient risk stratifications can be done, decisions support can be made. Okay, where the clinical decision supports can be provided, where we can look for the optimizing the hospital operations, right? Like we know that right now, I think many of you have definitely heard about, like we have the AI robots, right? Surgical robots are there. How? Okay, how the surgical robots are there, what exactly the technologies has been used over there. Okay, we are able to capture the real time data, that is called as the biosensors, right? Or as the smart mobiles, or as the watches what we are wearing. So ultimately it's capturing our real data every day, right? And it is suggesting you the how many steps you have walked, right? And it will even capture the data irrespective towards the patient phenotypic condition. So that is exactly what the enumerations means. The real data is get captured, and that makes the clinicians or as the hospital analysis to do a proper processing analysis, right? Predictive or as a preventive healthcare analysis, or as a medical image and diagnosis. This is a separate field, guys, where again AI is helping with respect to a disease or as the decision making finding. It's really a very exciting field. It's really a very comprehensive field. Okay, today we are talking about only multiomics, but apart from multiomics, we have separate domain area where the biomedical images, right, the clinical data, and looking for the earlier detection, precision medicine. So this kind of topics are even evolving. So you guys have to really remember this kind of concepts, that how we can capture those things. So ultimately AI is helping us to make a predictive modeling, guys. Remember, more we are looking for the prediction, because as we all know that COVID-19, the pandemic, the situation we all have observed, it's a real time, right? We have observed that. So now we are also looking that how predictive or as the future any kind of pandemic situation is getting evolved, so we can capture that based on the data. So this is exactly what the research is going on, and to do that AI is helping. So as I mentioned, that multi models are there, like generative models, right? So we are trying to use this kind of multi models where the seamless integration of multiomics data with the clinical integrations can be done. And why to do that? So that the onset or as the progression or as a treatment response can get monitored irrespective towards the clinical data, guys. Remember, all of you can see that on the screen, right? The image showcasing you the clinical data, it's getting integrated with respect to the molecular condition, and in that way we are able to make more predictive findings. So we have certain models, guys. Milton model success, like it predicts around 3213 disease phenotypic conditions, and it used the biomarkers and the plasma protein, which make a remarkable accuracy. You guys can really explore this. Okay, Milton model, you will definitely find a very fantastic researchers get captured where the remarkable accuracy, like they have identified the prognostic signature, where the outcome then leads towards the biomarker discovery with respect to the oncological targets and even with respect to the rare disease accordingly. And that is exactly what we really need to look with the disease area of our interest, right? And that's why you can see that the benefits of predictive analytics in AI or healthcare can be give for like patient care, personalized treatment, right? Population health management, because that is also really need to take care, right? Because we saw that during COVID-19, a bulk number of patient data is get captured. So how to use that data, how to store that data, that is also one of the challenging process, because the data once it get processed, right? So we the researchers who are working with this kind of things, we need to process and capture the findings. That's why we are capturing the data and we are trying to store with the proper management. Okay, risk analysis, right? Disease management, we can look for the healthcare digitalization or tracking, as I mentioned, the smart sensor, biosensors accordingly. Fraud detection, that is also one of the things which really need to take care with the healthcare kind of stuff, right? We need to even look towards how this the previous, I think I have mentioned about the Milton, enhance the power of genetic association study with the biomarker discovery, right? So that makes that the findings more what you can say is the real world. Okay, and that is exactly what this kind of findings is giving us a more boost to understand how the real world findings can be get achieved. All of you getting the insights, right? I hope so. The slides are helping you to understand the stuff, right? And I think we are very near to our conclusory slides, right? So as I mentioned that whenever we are talking about AI, big data, so ultimately today the era is trying to work all this processing with cloud base. Okay, so remember guys, whenever we are using this kind of big data, so ultimately today we have the AWS cloud environment, we have the AWS health omics environment, right? We have the Google cloud environment, GPU servers, HPC servers, where the ultra fast diagnosis can be done, single cell integrations of omics data can be done, and that makes a scalable infrastructure to process that data, such as the genome air horizon, which enables to make the diagnostic of 10 minutes per genome, guys. All of you are getting the point, right? So how this big data is helping with the cloud-based processing. Cloud security can be even done, guys, remember, because if you're working with the data which is get captured from the hospitals, right, the screenings are done, so security is also one of the concern point, and if that is even get cared by the certain cloud based platforms where the profiling can be done, integrations can be done, infrastructures where the proper data gets stored, managed, secure, and the pre-processing can be even done. Guys, are you excited for this, right? So this is exactly what is currently it's going on. Okay, and if you really want to be in that situation and you want to be in that race, then definitely you need to understand those aspects what we are today discussing. So some kind of case studies are even there, guys, so that you will understand how we can properly implement, such as AI driven insights in the biology, where the predictive analytics can be done. Okay, where we can look for the chronic disease management, earlier detections, improving the health management, right? Like, or else we are looking towards the enhancement of the cyber security, predicting the patient's preferences, handling the specific patient data accordingly. So many things deals with that, so it's really need to take care accordingly. So multiomics in AI or else the oncological with the precision medicine, why it's really important, because we are able to make the proper characterization or subtype analysis, as I think I have given you the example in my previous slides for the same, right? So the integration of AI will help us to decode this kind of tumor heterogeneity and understand the pattern and its correlation. And that's why AI is getting integrated with certain kind of fits. And if we go one step ahead, so it also have a clinical applications. Why? Because we can come up with the precision medicine findings. So that's why, guys, we need to come up with the clinical data integration, and we can even look towards the predictive or the prognostic signatures finding. Like AI has driven about like 9 gene per microRNA signatures, which predicts the high risk of the disease also, guys. And it will even show you maybe with adenocarcinoma or as any other disease, guys, you can pick up. It is giving you the clinical significance with respect to that. So it is not like that you're only finding the biomarker, but the biomarker, how it is relevant and shown a clinical significance, then it will give you a proper findings or the validations for your study. All of you agree with me? Yes. So we are not only taking the era about like AI, but we are trying to validate those things with the clinical significance. And that's make a very proper validatory findings with the integration of multiomics, AI, and the significant findings. Agree, all of you? Yes. And that's why you can see that the impact of rare diseases are even there, like the limitations. We are looking to addressing those kind of small patient cohort data, like population data, basically. Okay, where the certain kind of rare disease findings and the prognostic biomarker or the predictive biomarkers can be get identified, so that we can accelerate the proper pipelines for the drug discovery. Okay, and so that we can come up with the hidden analysis of the complex data, and that makes a success story, guys. Okay, where the neural network models, deep learning models are working to come up with a unpredictable details within the data, because deep learning or the neural network modeling what we have, it works based on the neural network capture. Okay, where we have the hidden layer, we have the proper encoder decoder, and it will start finding the correlations between the data sets, and that makes impactful and what you can say is a success story with AI integrations with omics data sets. The overall thing is that the takeaway things like the future of AI driven multiomics is transforming the healthcare, guys. Okay, And that's why today all of you are here to understand how we can come up with that understanding, like to get the proper overview. And that's why you can see, guys, AI evolution is going on. As I mentioned, generative AI, okay, automation process are going on. So how to work on the complexity of the data, how to use the proper machine learning architectures, the pipelines to understand the data. Real world insights is nothing but how we can move towards the actionable insights, that like a clinical decision support, which I've explained in my previous slide with the more validated study. Okay, how we can work on the cloud-based processing. So why cloud-based processing, guys? Because we are working with the big data. Okay, and it's really need to go with cloud-based processing. Then only the smoothness and the findings you guys can achieve. Okay, and that's why you guys really need to join this revolution. Okay, where the AI integrations and multiomics is getting a biomedical breakthrough, where you can make a key success in your career, guys. Remember, if you are a researcher, if you are a student, if you are a beginner, no matters. Okay, it doesn't matters at all. You are at the right platform today. Okay, so Biotechnica is a one-stop solution, as we all know. Okay, and that's why we have joined today. Okay, and we all of you know, guys, that ultimately today we are trying to know how we can move for the key to success, because it's unlocked the potential complexity of the molecular data, what we are trying to understand, right? And to make a decision, clinical decision making findings, transformative impact, because it makes a proper revolutionizing diagnostic or the prognostic treatment with the oncological disease or the rare disease. So ultimately the mission is on, guys. Remember, okay, the race is on, the mission is on, and you guys have to be in that mission. Okay, with the integration of multiomics data technology, big data, cloud-based processing is evolving, and you really need to come up with that findings, so that the advanced applications and the findings can be get achieved accordingly. And that is all about from my end. I hope that you all of you get the overview and the basic concept behind the integration of artificial intelligence, multi-omics screening, and definitely the more findings with respect to the big data analysis. So that is all about from my end. Thank you, guys.
Thank you so much, Dr. Nilofer ma'am, for beautifully wrapping it up with the computational and analytical dimensions. And you have connected NGS with multiomics and AI, and also showed us how big data is driving big discoveries. So your practical insights were very helpful. They highlighted how data interpretation and integration are real game changers in transforming raw sequences into meaningful biological breakthroughs, right? So yes, so it has been a very powerful session this evening, and in just 1.5 hours, we have traveled through from the fundamentals of sequencing to advanced analytics and precision medicine. And if there's one thing we've all learned today, is that the future of life science research belongs to those who can combine wet lab knowledge with data analytics skills. So sessions like this are more than just lectures. They are a glimpse into what your career can look like if you stay curious, skilled, and future ready. So that's exactly why Biotechnica continues to organize such expert led webinars to help young researchers like you gain exposure to real world tools, technologies, and mentors. And if you're inspired by today's session and want to build hands on expertise in sequencing and data interpretation, you must take up your next step by enrolling in Biotechnica's NGS internship program, which is starting from 15th October. And it's a guided project-based experience which is designed to help you analyze the sequencing data and understand the omics pipelines, even if you are a beginner. So as part of this internship, you will also work on real-time research projects that stimulate industry and academic workflows. So that will help you gain practical experience. It will strengthen your portfolio and also help you build confidence to handle real sequencing data independently. So don't just learn, start applying, right? Because that's where real growth begins. So we were awaiting for the Q&A session very eagerly. It's the time now for Q&A session. I would like to take up this one question from Sadaf Jan. So she mentions which generation sequencing is having highest commercial adoption or used for commercial scale. So Dr. Bhupender, would you like to take up this question, please?
Yeah, sure, ma'am. So, like the Urmi madam described, that Sadaf is interested in knowing that which particular generation sequencing is actually having highest commercial adoption. So, as per my understanding or the experience, I can say that second generation sequencing, especially the Illumina platforms, are have highest adoption because due to their say a balance in their accuracy and cost effectiveness and what you say data analysis part or computational resources. So one of the major, what I can say, major point that why they were so successful commercially, because the way or the read size which they actually give off is actually able to support the computational systems which were actually running through this throughout these years. But I can definitely tell that nowadays or in coming years, your third generation of sequencing will definitely capture the global market because as we know that those third generation sequencing are able to sequence very long reads, say in thousands of bases of long reads. But on the other hand, we also need computational resources for that to actually handle those data. So likewise, as the computational resources are developing, so slowly and steadily market will be captured by third generation sequencing based platforms. And but in maybe in other ways, if I would like to add one more thing. The adoption is one thing, and the other part is what type of analysis you are interested in. So if I talk about your human genome project, which was carried out, so that project was actually based on Sanger sequencing. Although they have done some major innovations in Sanger sequencing based platform, but that entire T2T sequencing was actually carried out based on Sanger sequencing. And if I say one more example, if I'll give you one more example, which I just shared during my presentation, which was of UK Biobank's whole genome sequencing. So they have adopted for Illumina platform. So what I want to infer here is this adoption also depends on the type of problem which you have in hand. But obviously, the bigger the size, the more computational power you have, you will definitely go with the third generation sequencing. But as of now, second generation sequencing, especially the Illumina platforms, are commercially successful in capturing the market. So I hope that will answer
Thank you.
Sadaf's question. Thank you.
Yeah, I hope so too. Thank you for the answer, sir. So I have a question from audience perspective. We have received it in our other platform. It's directed to Mr. Suhas, sir. So you have worked extensively in precision medicine for oncology and clinical genomics. What do you think, sir? How close are we to making NGS-based cancer screening a routine diagnostic tool in hospitals? And what would be the biggest challenge that will still remain?
I think so. We are working towards it. Maybe another, let's. I am hoping another decade we reach there because of the, like Dr. Bhupender said, the third generation sequencers and the cost going down. Because like if you compare to what it was costing to get a whole genome or even a targeted panel done, maybe 5 to 10 years back, the cost has maybe gone down by 50%. So cost is actually a bigger factor because when you come into the practical side of things, apart from the testing, they'll have other things that they need to take care of, maybe the clinicians and the drugs and everything. So a cost actually becomes a very big factor when it comes to sequencing and it becoming a routine. So at where I work in HCG, we try to integrate everything, and it is a holistic approach we take. So we have the histopathology, the IHC, and the genomics, and molecular biology, and cytogenetics. So we combine all that data for a patient to give. But again, it sometimes boils down to if they can afford. So an answer to that would be, and also in real world, it's also the availability of samples. Sometimes when it comes to it, the patient might not be able to give certain blood, or the biopsy might be very small in size, so you can do only one single testing. So I think so for it to become routine, we need to, and only thing I can think of is get the cost down. Everything else we are suited for because we have the expertise for it with and so many people looking into the analysis part of it. We have the computational power with AWS and other market players coming into it. So once we can get the maybe the the cost per sequencing, like now I think so from around a lakh or something, it's come to 50. Maybe we will go down to maybe where it's as affordable as a pizza. Then it becomes routine where you don't have to think of it like the way if you have ever been to a hospital, the first thing they write is a CBC. So it's a complete blood count. It's a very, very routine test. It's in the hundreds. So for sequencing to reach there, the potential is there, but I think so with upcoming technology and advancements, we will reach there, hopefully. I hope that
Thank you
answers
so
the
much, sir.
question.
Definitely that.
Yeah.
Yes, sir. And it will definitely make
Yeah.
our life a lot more easier. So with that hope,
Yeah.
let's take up one more question from Sujita. And I would like to direct this to Dr. Nilofer, ma'am. She is actually a part of bioinformatics internship in Biotechnica, and she's in confusion whether to go for NGS or AIML course. So she's interested more in NGS CRISPR techniques. So your suggestion, please, Dr. Nilofer.
Sure, ma'am. Yes, dear. So I
Yes,
think you have
yes.
already done the bioinformatics internship program. Then you are really aware about, like, we have covered more than 30 plus databases, tools, and softwares, and we have guided all of you guys with practically step by step. So might be like you got an overview, like what exactly we need to do with bioinformatics, right? And hand in hand, I think in with bioinformatics course, we have also showcased you about the NGS, that is next generation Galaxy software, right? And the pipelines for the same. And if you're really interested to deep dive with NGS and the CRISPR technology, so my recommendation will be like you just proceed ahead. Because as I mentioned with my starting of today's presentation itself, that if you're trying to utilize the artificial intelligence or machine learning kind of stuff, so it is an integrated way, like you're going one step advanced way of your finding. But prior to that, you should know the basic pipelines, that how exactly it's work. And if you want to expertise your thing with NGS pipeline and the CRISPR technology, and then come up with the integration, and then take help of AI for do the modeling or the predictive analysis, then I think that will be more impactful and more novel findings what you can achieve. So you just start, you have already done the bioinformatics, and just go ahead with NGS or the CRISPR technology kind of workshops, or is the courses what we have in Biotechnica, and then move for the AI. I think that is what you can move with your career. And all the best for the same.
Right. So there you go, Sujita. A sequence, Dr. Nilofer has suggested a sequence for you in your career ladder. Questions, but we are running short of time. So if you have any more questions, please write to us and we will definitely try to get back to you with the replies from our speakers. So before we close, finally, a huge round of applause for our distinguished speakers, Dr. Bhupender Singh, Mr. Suhas and Ghorpade, and Dr. Nilofer. Thank you so much for sharing your experience and expertise and making complex topics so understandable and inspiring. And of course, a big thank you to all our participants for being such an engaging audience tonight. Keep learning, keep exploring, and remember the next big breakthrough in science might just come from you. So, thank you, everyone. With that thought, have a wonderful evening ahead. Bye bye.